So hello everyone. Today I would like to discuss with you susceptibility because we are all exposed to all kinds of chemicals and we can react to those compounds in different ways, right? Some people get a disease very early, some others get a disease very late, although they are exposed to the same amount of compound, right? So susceptibility plays an important role. And also in regulatory toxicology, we take this variability into account, right? Because with the uncertainty factors, we take human variation or human variability. We do take that into account. We have a factor 10 for this uncertainty factor because we want to protect the most sensitive persons in a population. Of course, more research in understanding why some people are susceptible and others are not can also more refine these uncertainty factors. So what I would like to discuss today with you is I would like to first discuss some subgroups that are more susceptible. I want to talk about variation between humans in the metabolic activation and detoxification, and how that makes you more susceptible to certain exposures. I want to talk about polymorphisms, genetic polymorphisms for carcinogen metabolism, because polymorphisms may be, at least in part, explain the variation that we see. We can of course wait until people get cancer, but usually we want to know results earlier, and that's why we use intermediate markers in human studies. And I would like to end with some concluding remarks. So let's start with susceptible subgroups. So there are various susceptible subgroups, subpopulations in a total human population, and what you can think about is, for instance, children. So children are also exposed to the same air, the same food as we as adults are, but of course because they are smaller, the dose per kilogram body weight is of course much higher for children as compared to adults. On top of that, children are still developing. And for instance, the brain is developing, the heart is developing. So while exposure while you are developing can also have an impact, a higher impact on the organ than in adults. And we also have that children have different habits. Think about that children put all kinds of things in their mouth, and then that may also lead to additional exposures. So if then think about development is also in the womb, of course. So if you are a pregnant woman, also your physiology changes when you're pregnant, but also the fetus is also developing. So that's also a very sensitive population. Next to very young people, like the children, also elderly people may be more sensitive, because of the biological changes that occur when you are aging. Another susceptible group could be immunocompromised individuals, right? They have a weakened immune system, and therefore they may differently react to all kinds of exposure, particularly when you think about pathogens like exposures to viruses and bacteria, etc. And then we also have differences between populations with regard to the level of exposure. So people that are working in a specific area may be exposed more to a certain compound than people that are not working in that same field. And therefore, occupational exposures may also make some people more susceptible. And there we also have an effect, what we call the healthy workers effect. So also, people that are exposed at work to high concentrations, if they can stand that exposure very well, they continue working in that field or in that factory. Whereas if you are more susceptible, you get sick very early and then you drop out as a worker, right? So the people that work in certain factories may also have a different type of susceptibility or vulnerability as compared to people that drop out. So these were all kinds of different susceptibilities and populations that are susceptible. But even within the normal general population, we know that there are people that are more sensitive and people that are less sensitive. Right? So we have an exposure, and eventually that leads to cancer. But we all know that if I take smoking as an example, we all know that there are smokers that get cancer at a very young age, but there are also smokers that do not get cancer at all, and then they die from something else. And this susceptibility, so this vulnerability for this exposure, in this case cigarette smoke, can have a genetic background, so it's your genetic makeup, but it can also be a acquired susceptibility, which comes from, for instance, your lifestyle or your behavior, etc. The genetic susceptibility, that cannot be modified, whereas your acquired susceptibility, often that can be modified because you can change your behavior, you can change your exposures, etc. And your diet, for instance, can be changed. Right? So on one hand, you have the genetics, which cannot be changed, non-modifiable. On the other hand, you have your lifestyle that can eventually be modified. So this brings us actually to the idea of how susceptibility plays a role. So if you have no exposure, your risk level will be low, independent of what kind of susceptibility you have, right? Because your exposure is still the most important determinant of getting the disease, yes or no. Whereas if you then think about people that are exposed, you can have people with a more resistant genotype. So they still have an increased risk because they are exposed, but the people that are sensitive, so for instance, because of their genetic makeup, they have an increased risk when you compare to those which are more resistant. Both groups have an increased risk because of the exposure, but there is a difference between people in that population depending on whether they are more resistant or not. So genetic variation may be the basis, at least in part, for this variation in susceptibility of individuals. And there is a lot of different types of genetic variation. We have, and the most important one, I think, are the SNPs, the single nucleotide polymorphisms. And they are very common in a population, right? So they are, by definition, more than 1% of the individual have certain SNPs, otherwise they would not be called a variant, but rather a mutation. And these SNPs, these genetic polymorphisms, they can also be in genes that encode for carcinogenesis related enzymes. So how can you imagine then how genetics play a role? And in the beginning, you can see the first one indicates that if you have a SNP in a codon region of a gene, then that may lead to a change in the amino acid that is encoded in the protein then. And then if there is a change in the protein, you may have a different activity of that protein, making somebody more susceptible, yes or no. There can also be a deletion, right? So a SNP would lead to an altered amino acid, but if you have deletions, then of course a whole part of the enzyme can be absent, and then of course you also don't have enzyme activity. But also polymorphisms in non-coding regions can be important, because polymorphisms in non-coding regions can affect, for instance, transcriptional control. If you have a polymorphism in the promoter region where transcription factors should bind, and the transcription factor cannot bind or can bind in a lesser extent, then you of course also affect the transcription of that gene, the amount of protein, and therefore often also protein activity is affected. You can also have genetic polymorphisms that increase the half-life of the mRNA. So with one single mRNA, you can make multiple proteins. And if your mRNA lives longer, you can make that same protein for a longer period of time. Gene amplification is a next example. If I have two copies of the same gene, I can transcribe it twice in the same period, in the same time amount of time, and therefore I may have more enzyme activity in my cells. On top of that, there is of course also a interaction between the different genetic polymorphisms. So the endpoint, the actual effect that we have is not always clear from purely genetics. I will come back to that in a later slide. So there are a lot of examples of diseases that are clearly linked to a single genetic variant, right? So cystic fibrosis, you have a mutation in a salt transporter, and that leads to that disease. But that is one gene leading to a disease. But when we talk about genetic polymorphisms and susceptibility, it's much more, let's say, multigenic. There are multiple genes involved, and therefore the penetrance will be lower, right? So in a single gene disease, one mutation leads to the disease, whereas and that is a very high chance of getting that disease. Whereas if you think about the susceptibility genes, you need exposure, you need multiple genes to be defective before you eventually get a result. Right? So environment plays a big role there. So if I would then compare, let's say, the single gene diseases and the diseases that make you more susceptible, also leading to a disease, is that the frequency of the single genes is rare. Usually they only occur in families, right? So they are inherited from father or from parents to the children, but then only within that family, but the general population is not really affected. And these are studied usually by linkage, right? So because you see how they are inherited from one generation to the next generation. The penetrance is high. So if you have the mutation, you will have a high chance of getting the disease. And that's also then a high absolute risk. But because the mutation is so rare in the population, there is a low attributable risk. And that is of course different in the susceptibility genes. They are very common. They are more than 1% of a certain polymorphisms occur in the population. They are studied by associations often, but the penetrance is low, right? So just having that gene is often not enough, or that gene variant is not enough to get a disease, right? So usually also the environment plays an important role, right? So the role of the environment is critical in susceptibility, in these types of susceptibility genes. So the risk is low, but because they are so common in the population attributable risk can still be quite high. And these genetic polymorphisms, they occur in all kinds of different genes that are important for how we deal with xenobiotics, how we as humans deal with exposures to these foreign compounds. Right? I just have a list here. I will explain a little bit more in this presentation about cytochrome P450s, glutathione S-transferases, and myeloperoxidase. Because there is a genetic determinant in the variation of enzyme activity, it would be very logical to do genotyping. Just look at the genetic variant to predict what is going on with enzyme activity. And in some cases, that is really possible, right? So GST-M1, glutathione S-transferase M1 is an example. There is a deletion of that gene in 50% of the Caucasian population. If you don't have the gene, you will not have the enzyme activity. So you can already, at the DNA level, predict what is going on on the enzyme activity. Whereas, for instance, cytochrome P450-3A4, a very important metabolic enzyme in the liver, that is really also affected by your environment, exposures. It's inducible, right? So there, the actual activity is not only determined by the genetic polymorphism. So there, it's not wise to do only genotyping, there it would be much better to look at protein activity. So some examples of polymorphic variants in detoxifying enzymes and how they are related to health risks. So the GSDM1, which I already mentioned before, is deleted in 50% of the Caucasian population. That leads to that you don't have an enzyme, increases your lung cancer risk. GST, very similar. There is a deletion leading to, for instance, colon cancer and astrocytoma. Epoxide hydrolase, there is a variant, a mutation, a T to C mutation in the DNA in the coding region that leads to decreased activity, and that increases, for instance, your risk for COPD and emphysema. Myeloperoxidase, here the genetic variant is located in the promoter site, right? So the transcription factors can no longer bind, and as a result of that, you have reduced transcription, reduced enzyme, and therefore you lower your risk of lung cancer. I have an example of that later on in this presentation. And others are GPX, it's related to lung cancer, and so therefore this is these are examples of how these genetic polymorphisms can affect your cancer risk, for instance. I already mentioned that you can do genotyping. In some cases, this genotyping can be very simple. For instance, the GSDM1 again, glutathione S-transferase, it's a deletion, so you can just look for the deletion, very simple analysis, and you're done. Whereas if you go more to the right, the NQO1, for instance, there you have a lot of different genotypes. Right? So it's not only one variant, but multiple variants in your DNA that determine what your phenotype eventually will be. So here you could choose, of course, for genotyping, but it's much more complex because you have to look at multiple loci within the gene. So it's not so straightforward often. As I also mentioned, is that environment plays a critical role, because you can have that variant, but if you are not exposed, you will not have any effects of that variant, right? The exposure is very important. So why do we want to study these gene-environment interactions? First of all, we want to understand more of the exposure disease process, right? By understanding why somebody is more susceptible than another one, that can also increase our knowledge about the whole process, right? We also want to understand why are there susceptible subgroups, because they need to be protected more than the non-susceptible groups. We want to prevent adverse drug effects or drug diet interactions. They can also be related to your genetic makeup. But then also, as I said, in the risk assessment, we now have an uncertainty factor of 10. And maybe we can improve or refine that uncertainty factor in some cases to a larger degree, right? And because we have these susceptible subgroups, they need to be protected as well, right? And therefore you could target prevention strategies specifically to these susceptible subpopulations. So this is why we would like to study these gene-environment interactions. But again, it is important to note that the environment is still the most important cause of the disease that you are studying. So genes load the gun, but a loaded gun is not killing you. Only if you pull the trigger, then you have the problem, and that trigger is pulled by the environment. So the next part, I would like to give you also some examples, right? So the whole idea here is that if you are exposed to a carcinogen, usually these compounds are not yet mutagenic or genotoxic by themselves. They need to be metabolized. And this metabolism usually can lead to excretion in the urine, but occasionally you can also get reactive metabolites during your metabolism. These can bind to DNA, and that leads to cancer. And whether your genetic makeup can determine whether you are more on the left side of this graph or on the right side of this graph and having an increased risk for developing cancer. An example of that is Winston Churchill, right? He smoked a lot. Actually, on almost all pictures that people took from Winston Churchill, he is having the cigar in his hands. I think there was only one picture without a cigar, and that picture is actually known as the picture without a cigar. But still, Churchill, although he smoked a lot, he drank a lot, had a stressful life, he became 91 years of age, right? So he was probably genetically more on the left-hand side of this graph. He excreted the metabolites instead of making them more dangerous. And there are different interactions in this as well, right? So this is, for instance, a relationship between the phenotype of cytochrome P451A2 and N-acetyltransferase. One is activating the compound, the other one is detoxifying the compound or also activating it, right? So, and then if you see the high phenotype and the rapid acetylators, they have an increased risk for when they eat well-done meat, right? So the exposure in this case is the well-done meat, and you see that there is also a combination effect of the different genotypes that are in the same pathway, right? So they increase their risk or their effect because they are in the same pathway of the process, from the compound itself to its reactive compound. Is it really that simple? Unfortunately, no. In the beginning, when these genetic polymorphisms were discovered in the 1980s, 1990s, we thought, okay, now we have the Holy Grail. Now we can explain why some people are more susceptible than others. And the original papers that were published, they had really very high odds ratios that were published, right? So a lung cancer risk of 10 or something like that, right? So a relative risk of 10. But then when more papers were published, you also had a lot of negative data. You see that the impact of single genes, single genetic polymorphisms, became smaller and smaller and smaller because you have an accumulation of the evidence, right? And that already indicates that you are dealing with complex diseases. So and genetic susceptibility genes, it's not a single gene disease, it's a multigenic disease. So you need more than simply one single genetic polymorphisms to explain the susceptibility of an individual, right? So there were some confusing results. If we look for the single genes, I think the glutathione S-transferase as an example, it is still statistically significant. The effect of GST-M1 on lung cancer is still statistically significant, but the relative risk really decreased over the years because you also had the negative papers being published, and therefore this effect of this one single gene did not really matter anymore, or was less pronounced. So, but then even within this group of people with just glutathione S-transferase deletion, even in that group, you could maybe have subgroups that are more affected by this, by missing this gene than others, right? So even within the susceptible group, you have people that may, because of additional factors, are still very even more susceptible. So that's where molecular epidemiology comes in. We have the exposure, we have the disease. We see that some people go very quickly from one end to the other end. But what we want to do in molecular epidemiology is we want to break open that black box in the middle, because we want to understand why is somebody going very quickly from exposure to disease and the other one is going to going very slowly towards the disease, so it's more resistant. And we do that, or we can do that, by measuring inside a human being, right? And one of the most important measurements in the past that we used is what we call the biologically effective dose, which is the DNA damage, for instance. Right? So, and the susceptibility factors, they actually determine how quickly you go from left to right in this graph. And a lot of studies that we did to understand why some people are more susceptible than others is done in cigarette smokers. And the reason for that is because we know that cigarette smoke contains a lot of carcinogens and genotoxic compounds, right? So we know these people are exposed, and it's very easy to determine how much these people are exposed. You can simply ask, how many cigarettes per day do you smoke? Or you could actually measure cotinine levels or nicotine levels in biological fluids, right? So the cigarette smoke contains a lot of DNA damaging compounds, and therefore studying cigarette smokers is a very nice thing to do to understand the underlying mechanisms. So the study that I would like to show you here is a study on 67 smoking volunteers. We had blood of those volunteers. We measured, on one hand, we measured the DNA damage, and we measured a lot of genetic polymorphisms in xenobiotic metabolizing enzymes, so the enzymes that are needed to metabolize the compounds in cigarette smoke, and that leads to DNA damage. So this is the whole list: cytochrome P451A2, glutathione S-transferase, which I mentioned already before, but also some DNA repair genes like XRCC1, XPD. We also had some detoxifying enzymes like glutathione peroxidase. So this is the whole list of genetic polymorphisms that we determined in these individuals. And we wanted to see how is the relationship between the cigarette smoking and the DNA adducts, the DNA damage, and how is that affected by these genetic polymorphisms. And what we then see is that when we look at all those genes and we sum let's say, and for each gene, you could say, okay, this variant is the good variant, the other variant is the bad one. And then if you would sum up all the bad alleles that somebody has, we see indeed that the people with more bad alleles in all those genes, these were the ones with higher levels of DNA damage. Even though, if you this on the right in panel B, you see the amount of cigarettes smoked per day by these individuals, there is no difference in the amount of cigarettes that they smoke. So this is from the whole list. So from that, we looked for statistically which one were contributing the most. Then we came up with GST-M1, epoxide hydrolase, and glutathione peroxidase. And when we then look at the sum of risk alleles in these individuals, again, those that carry more bad alleles, bad variants of those genes in their DNA, that were the ones with more DNA damage compared to the ones that did not have those bad alleles or having the good alleles, I would say. Again, cigarette smoke exposure, so from self-reported amounts of cigarettes that they smoked, there was not different between the people. So exposure is the same, but the adduct levels are completely different between the susceptible groups and the non-susceptible, and the susceptible groups on the right and the less susceptible people on the left. So then DNA adducts, that's nice, right? But you also would like to know what happens to cancer. And also here we looked at a study in which we took we investigated this squamous cell carcinoma risk. And what you see is that the most susceptible people, in this case, a combination of a genotype in glutathione S-transferase and cytochrome P451A1, that the people with the bad combination of the alleles are also the people with the highest risk of squamous cell carcinoma. But these was also the people with the highest levels of DNA adducts in both their white blood cells and in their lungs. So on one hand, we have this genetic susceptibility, but in the beginning of my talk, I also talked about acquired susceptibility, right? So things that you can change, right? So diet, for instance. And of course, there is an interaction between your genetic makeup and your acquired susceptibility. And this is very nicely demonstrated in this study by Pali et al. If you look at the people that do have the glutathione S-transferase M1 gene, right? They do not have the deletion. If you then measure DNA adducts in those people, you see that all kinds of dietary habits or dietary intakes, and these are thought to be healthy dietary intakes, there is no difference or not really a difference in DNA damage on basis of their intake. Right? So low, medium, high intake of, for instance, vitamin E, beta carotene, fish, raw vegetables, cooked vegetables, leafy vegetables, etc. No relationship. However, they do have the GSD-M1 gene, so they are already protected genetically. So then they looked into the people that missed the GSD-M1 gene. So these people actually need additional protection because they already lack the gene that protect them, right? So, and what you then see is indeed that a high intake of, for instance, vitamin E or leafy vegetables or vegetables in general protects those people, right? So your diet is protective only in those people that are genetically susceptible, right? So there is also an interaction between your genetic susceptibility and your acquired susceptibility. So your lifestyle, your diet, etc. The next example I would like to give is myeloperoxidase. So myeloperoxidase is a gene that is an enzyme that is present in neutrophils, in inflammatory cells, and it is released as a defense against microbes. Right? So if you have an infection in the lungs, for instance, myeloperoxidase is released into the environment to kill those bacteria. But it's also able to metabolize benzoaperine. Benzoaperine is a very important cigarette smoke derived carcinogen. In myeloperoxidase, in the promoter region of myeloperoxidase, there is a genetic variant, a polymorphism, that can make the gene less active, right? So you have less transcription of the gene, therefore less protein, and therefore lower activity of the protein. So the people with the mutant AA alleles or the AA genotype, so both alleles are carrying the low activity alleles, they are known to have a lower lung cancer risk. But again, myeloperoxidase, as I said, can help in the metabolism of benzoaperine. So that's why we looked for DNA adducts in those individuals. And we expected that the low activity alleles will have lower DNA adduct levels. Because if it is involved, especially in this second step here, in forming the active metabolites of benzoaperine, if you have a low activity, you will also get less amounts or lower concentrations of your active metabolite, less DNA damage, lower lung cancer risk. That's the whole underlying idea. Right, so here, this is then a little bit graphically. So if you have the high activity allele of MPO, you will have more DNA damage because you activate your benzoaperine and therefore a higher lung cancer risk. Whereas if you have the low activity alleles, you will have low DNA damage and lower lung cancer risk. Right? So that's the whole idea behind it. So we had access to lung lavage cells, so cells from actually coming out of the lung during a bronchoalveolar lavage. We had cells, we had fluids, so we measured cotinine levels to show that they are really smokers. We determined the MPO polymorphisms, and we matched the people between the different genotypes, and then we measured also the DNA adducts, so the DNA damage in those cells. And so here, first of all, we showed that the genetic polymorphism is really, let's say, active, because the high activity MPO people, so that were homozygote for the high activity allele, they indeed have a higher MPO activity in their lavage fluid compared to those with the low activity alleles. Right? So the genetic polymorphism is actually determining the phenotype with regard to activity of the enzyme. As I said, we matched the people with the different phenotypes on basis of, or genotypes, I mean, on basis of the sex, the age, the amounts of cigarettes that they smoke, the tar per cigarettes, etc. So we had 11 people matched for all the 3 genotypes. And what we then see is that indeed, we measure DNA damage in the lung cells, or in the lung-derived cells, I have to say. And we do see indeed that the ones with the high activity alleles have more damage compared to the ones with the low activity alleles. On top of that, that is really interesting. If we looked at the dose-response relationship, so we looked at how many cigarettes do these people smoke and what is the amount of DNA damage. In the overall group, we did not see a very nice correlation between the exposure and the amount of DNA damage. However, if we split this group up on basis of the genotypes, what we then see is that the relationship between this exposure and the damage is much higher in the ones that have the high activity compared to the people with the low activity. So at the same dose, the people with the high activity alleles make more DNA damage, and therefore they have an increased risk for developing lung cancer. So this is then genetics, your diet, etc. But susceptibility, as I said, also your child is developing, and now recently there's also more interest in other sources for individual susceptibility. And one of those new sources or new ideas is what we call the developmental origins of health and disease, the DOHAT theory. Right? So the child in the womb is exposed, it will adapt or it will respond to that exposure. It will respond to, for instance, also to the maternal diet and maternal lifestyle, etc. And then after being born, that adaptation is more or less fixed, and you will carry that with you for the rest of your life, and that leads then to certain diseases, being more susceptible for certain diseases, yes or no. Very nice example is the Dutch famine, what we call the hunger winter at the end of the Second World War. The food supply to the rest of the country of the Netherlands was cut off, so therefore people, even those people that were pregnant, could not eat enough. The baby adapted to that. After the war, they could eat as much as they want, and what we see is, due to this adaptation, we see increased levels of obesity in that population. Right? So the baby is adapting, and it can adapt to food triggers, environmental triggers, exposures to compounds, etc. So it's much more complex even than what I explained so far. So again, to wrap up, to come back to my original slide, in regulatory toxicology, we do have these uncertainty factors to, let's say, extrapolate the value or the exposure that we still allow. And these uncertainty factors are, let's say, a guess. We assume that, and then we often have a factor of 10. We assume that the difference between the most and the least susceptible individuals is a factor 10. And I think that can be refined even more, because in some cases you may say, okay, 10 is too high. In other cases, you may maybe say that 10 is not high enough. Right? So there needs to be some refinement on these assumptions, I would say, and this type of research can help in that. So in conclusion, we are all different, and these differences in susceptibility can have multiple origins. We can have a genetic origin, we can have let's say, age, sex, lifestyle. I didn't even talk about all of the factors that determine your susceptibility, but for instance, age and sex is also an important one. We study DNA adducts usually as a very early endpoint to study these genotype-phenotype relationships, to understand susceptibility, because we don't want to wait until it's too late, right? Because if you, in your studies, you can wait until people get cancer, but of course then you are too late. We want to see what's happening in the early phases, right? But due to the complex exposure, I think you now understand that susceptibility is very difficult to understand. The underlying mechanisms that makes you more vulnerable to certain compounds or certain exposures than others is very complex. It can be genetic, even in combination or with interaction with the genetic and the acquired susceptibility.